is procyantine. Procyantine, which is an anti-tumor compound, which made it to phase 2 clinical trials. It has been abandoned afterwards because it proved to be too toxic at that point. But I think there are currently efforts to revisit this compound and maybe look at related compounds and to go into deeper drug development of these metabolites. And then one compound which is very interesting for us for ecological reasons is ailanthone, which has allelopathic properties. And what allelopathic means, we'll talk about in a second. So ailanthone is made by Ailanthus altissima, the tree of heaven, which is basically originally an ornamental tree, but right now I think it's mostly known as a globally invasive plant. In Europe, it was originally introduced as an ornamental tree, but right now it's found on all continents except Antarctica, so it's basically everywhere. I've also seen this in Canada during a visit last year, so I definitely know that it also exists on other continents, but especially in Germany, I also know that it basically exists everywhere. I think this is one of the plants, once you get to know them, you can really basically spot them everywhere. So I think this really shows how important it is to train your botanical eye and look for plants. And I'm pretty sure if you start to know this plant, you will also be able to detect it everywhere in your neighborhood and wherever. Growing out of concrete and especially along railroad tracks, it's really widely invasive. And due to this invasive properties in the European Union, this is now part of a list of invasive alien species of union concern, which means that all European Union member states need to take active measures for preventing the further spread of this species, which definitely proves that this is a species which has some very big ecological impact. Now, coming back to the quassinoids, one phenomenon that you can see very often in nature is that if you look at the growth zone and below these Ailanthus altissima trees, is that you can actually spot that there's very little other plants growing. And there have been some very nice experiments in the 90s by Robert Heisey, and he figured out that these so-called allelopathic properties are mostly related to quassinoids, and especially this most active compound, ailanthone. Allelopathic basically means that these compounds interfere with other plants growing or other organisms, to be more general, growing in the environment. So he did a couple of experiments. Here you can see the application of ailanthone post-emergently with increasing concentrations. So basically after other seeds have germinated, or alternatively, he also did a series of experiments where he added ailanthone before other plants emerged. So you can see that ailanthone is able to prevent the germination of other plants in the surrounding. These are the controls, so this is how it should normally look like. Again, control. And after the germination, they basically also inhibit the growth of other plants. So obviously, these quassinoids have a huge ecological relevance as well. This is, of course, very interesting, not only for us, but hopefully for many other people as well, because, yeah, there's this big ecological relevance, but also there are many potent other biological activities, for example, these anti-tumor properties, but also for insecticidal activity, many quassinoids could be very interesting. The biosynthetic pathway towards quassinoids so far is basically completely unknown. And of course, for us, this is a great chance to do some work in this area. Because additional knowledge about the biosynthesis would not only be relevant to find out more about these interesting structures, but also to shed light into the ecological relevance and at some point to maybe use biotechnology to produce these compounds in a better system. So quassinoids are structurally really unusual. They are actually triterpenoids, and I guess all of you know very well that triterpenoids by definition are derived from 6 isoprene units. They have 30 carbons, but this is actually not true for the quassinoids because they only have 20 carbon atoms, at least most representatives. So they are actually called decanor triterpenoids because they lack 10 methyl groups, so to say, or 10 carbons. So really unusual, but there has been some very solid feeding experiments in the past. So the fact that they are really triterpenoids is very firmly established. So somehow during the biosynthesis of these 30 carbon molecules, 10 carbon atoms have to be lost again, which is very unusual and I think a very interesting fact that we are working towards. So quassinoids are not the only unusual triterpenoids in the order of producing plants. The family Cimarubaceae belongs to the order Sapendalis. And there are two neighboring families in the order Sapindalis, the Meliaceae and the Rutaceae. And both of these produce another class of related triterpenoids with unusual structures, the so-called limonoids. Limonoids also are triterpenoids with less than 30 carbon atoms. They have typically 26 carbon atoms, so they are called tetranor triterpenoids. And two very famous limonoids are azadiractine, which occurs in chinaberry, Meliazadirac, which is a very potent insecticidal compound. So if you've ever seen or heard of neem oil, which is one of the strongest insecticidal compounds or extracts that you can find out there, the main active ingredient of this is azadiractine, a very complex molecule, obviously. Another very interesting molecule is limonene, which occurs in very well-known citrus fruits like orange and lemon, for example. This also has some bitter properties. I guess many of you have already noticed that sometimes orange juice can have a certain bitter taste, and this is partly related to the presence of limonene. So also flavonoids occur in citrus fruits, which have some bitter taste, but limonene definitely contributes as well to the bitter taste of citrus products. So obviously there's also a big relevance of these unusual molecules. So in the past, people have actually proposed that there might be a common biosynthetic origin of limonoids and quassinoids. So if you look at the structures, there are, of course, pretty major differences. But if you look at the carbon skeleton and use your imagination a little bit, there are definitely some similarities in the carbon skeleton. So I highlighted here the main ring structures. And if you look at the positions of the methyl groups, the stereochemistry, for example, you can actually see that it does look a bit like they are related. So in the past, this has been only proposed on the structures and some biosynthetic predictions, but there has been no evidence so far that really limonoids and quassinoids are related. But of course, considering that they are in sister families, I think this is a very intriguing hypothesis. When we started our work on the quassinoids, the first three steps of the limonoid biosynthesis had just been found out and published by collaboration, very nice work by the Osborne and Satterley groups. And they discovered that first there's an oxidosqualene cyclase step, which makes tirocaladeinol. And then there are two P450s, which convert this basic carbon skeleton into two modified versions, so-called proto-limonoids, dihydronilotizine and melianol. So this was the state of knowledge for the limonoids when we started our work. For the quassinoids, not even a single biosynthetic enzyme was known before when we started our work. So our work and our aim still is really to investigate the biosynthetic pathway and really discover how these intriguing carbon molecules are made up. For this, we use a workflow which is probably very familiar to many of you. And I think there have been lots of presentations in this very nice seminar series, which use a very similar approach. So basically, we first start with plant tissues and then use RNA-seq to generate transcriptome data and look at the spatial expression patterns of different gene candidates. And then we basically use co-expression analysis to use and express the gene candidates and then perform some biochemistry. And we use also the very well-known system by transient expression in Nicotiana and Bentamiana to probe the function of gene candidates. Because in many cases, we have 20, 30 gene candidates and we need to test many different combinations. And for that, of course, transient expression in Nicotiana and Bentamiana is really a fantastic system. I think many of you know this very well, but I will still give a very quick introduction because the very important point about the system is the fact that you can so easily co-express multiple genes. For example, in our case, for triterpenoids, we not only look at gene candidates, but we also can very easily include some booster genes, which basically encode for enzymes from the mevalonate pathway to boost the precursor amounts of oxidosqualene, which is the first intermediate in triterpenoid biosynthesis. So all of these genes are closed into expression plasmids and then transformed to Agrobacterium tumefaciens for transient expression. We just mix the different strains. I think you all know this very well. And then we normally, for test reactions, we use syringe infiltrations to test different gene combinations. And then we extract the metabolites, do some LC-MS or GC-MS analysis, and then hopefully we see some new activity. So one of the main challenges of our work is that for quassinoids, there's basically no standard available at all. So every time we see some new peaks, we really need to figure out what these peaks are, which basically means that we need to purify compounds and use NMR analysis to figure out the structures. So it's very clear that very often we need to do large-scale experiments, and for that we've improved our system for doing these transient expressions in larger scale a little bit. So I think many of you know that there already have been some very nice solutions in the literature, but many of them use very complicated equipment, which is very difficult to achieve if you don't have very high engineering skills or some custom-made equipment. So in our setup, we basically use two different desiccators where we basically can alternate between the two desiccators, so we can prepare 3 plants already for the next desiccator while the vacuum infiltration is going on for the other one, and then we really just move the whole cap to the other desiccator, which is extremely efficient, so we don't have any long waiting times in between. And for holding the plants in place, we've developed some 3D printed plant holders. This is actually a very low-tech solution, but I think these work very well. So these are basically just some plastic discs that you can slide over the plants, and they don't have any sharp edges. They fit perfectly, hold the plants in place, and you can take them off and put them on again very quickly. So this has been really nice and efficient to improve our workflow. This was a very nice collaboration with the group of Janina Bahnemann at Leibniz University, Hannover, who is an expert in 3D printing, and this really made the system much easier. So with this, we can typically achieve infiltration rates of around 40 plants per hour. And for most applications, we need something like maybe 80 or 100 plants, and therefore it's definitely possible to do this in something like an afternoon, I would say, to get enough material for NMR analysis in the end. All right, let's come to the real biochemical work. Of course, when we suppose that these compounds are triterpenoids, the first step that we are looking at is, of course, a cyclization step catalyzed by this big enzyme class called oxidosqualene cyclases, which are really nice enzymes which take oxidosqualene, a more or less linear precursor with the epoxide ring here, and then form typically a tetra or pentacyclic skeleton out of this. So if these quassinoids are really triterpenoids, of course, there should also be an oxidosqualene cyclase doing the first synthesis step. So we looked at our transcriptomic data that we generated for Ailanthus altissima, looked at expression pattern, homology to other known enzymes, and in the end we found 3 candidates where we really couldn't be sure what would be involved in quassinoid biosynthesis. We called these enzymes AAOC1, 2, and 3. All of them had some decent expression in the bark, which is one of the major tissues where you can find quassinoids. AAOC2, in addition, also had very strong root expression. They all had some very decent overall expression. So we couldn't really be sure which of these would be involved in quassinoid biosynthesis. So we went ahead and expressed all of them in Gutiana bentamiana, and so here you can see just an empty vector control. This is empty vector with the booster genes, which increase oxidosqualene, which is this peak here, and this mostly makes cycloartenol, which comes from normal phytosterol biosynthesis. And here you have the co-expression in addition to the boosters of AAOC1, 2, and 3. So for OC1, it was very complicated, and we still are not really sure what it does because we just obtained multiple peaks in low levels. So unfortunately, we have not been able to isolate any of these structures or compounds in sufficient amounts for NMR analysis. So we still don't really know what this does. For OSC3, in comparison to an authentic standard, we could clarify that it's a loopule synthase. And for AAOC2, if you compare to the booster control, it looked like it didn't do anything. So obviously this was at first pretty disappointing because it looked like there was no clear new peak related to quassinoids. But then we looked closer at the mass spectra of the AAOC2 producing strain, and this was really surprising to us because even though the peak looks perfectly symmetrical and it really looks like there's no change at all, in the mass spectrum we really saw a clear difference. So here you can see just the mass spectrum of cycloartenol. This is what we have if we only have the booster genes without AAOC2. And you can see the mass spectra are virtually identical between cycloartenol and the boosters. But if we additionally co-express AAOC2, we get a very strong shift in the mass spectrum. It looks very different from the normal background control, which told us that something has to be happening, even though from the chromatographic analysis, there's virtually no difference. So we went ahead, did a large-scale infiltration, isolated the compound, and we could really confirm that it's tyrocaladienol, so the same precursor which has been found in limonoid biosynthesis. So we went ahead and looked for additional steps in the pathway. We also assumed that there should be some additional oxidations happening. So we looked first at cytochrome P450 monooxygenases, which are very broadly involved in tritopy metabolism, calculated the Pearson correlation coefficients to this oxidosqualene cyclase, and then looked for the genes with the highest co-expression and also very high total overall expression. The total expression is shown here. And from this, we ended up selecting 10 candidates, of which we could successfully clone 8. So what you can see here is also the weakness of these correlation coefficients. If you use the simple Pearson correlation coefficient, because it really turns out that there are two patterns emerging. So the oxidosqualene cyclase has a strong expression in roots and in the stem bark. And you can really see that there seem to be two different classes of expression patterns with high Pearson correlation coefficient. So there's one class which also has the same strong bark expression here and here, but maybe not so much root expression. And then there are also some very strong root expressed candidates, which maybe not necessarily have stem bark expression. So this definitely showed us that maybe the Pearson correlation coefficients would not be very efficient for later work, but I'll come to that in a second. Nonetheless, we went ahead with what we had so far, and we cloned the gene candidates, did some co-expression again. And here's an example of what it looked like normally for most of the candidates when we had co-expression of the OOC2 with the P450. We got the small and not very nice background peak of tyrocaladienol, but no new peak. But only for one of the P450s, we got a new peak with an increased mass of 32, corresponding to 2 oxygen atoms. This was very nice to see. And then when we co-expressed this CYP71CD4 with an additional P450, we got yet another new mass shift of +14, so +1 oxygen and -2 hydrogen. This led to another compound which we isolated and confirmed by NMR that it's medianol. So the same compound which also has been found in limonoid biosynthesis. So to wrap up this part of the story, we could really confirm that the first 3 steps of quassinoid and limonoid biosynthesis are really biosynthetically and evolutionary related. For each of these 3 steps, there's a related homologous enzyme. So first, the homologous oxidosqualene cyclase. Then there's a CYP71CD P450, and then a CYP71BQ P450, which convert tyrocaladienol to dihydronylotysine and medianol. So it really seems like medianol is a conserved key intermediate, not only for the limonoids, as was shown before by N. Osborne and Beth Setterly, but also for the quassinoids. Which is definitely a bit surprising because as I said before, both of them are very heavily truncated tritopenoids. For the limonoids, just these 4 carbon atoms in the side chain are cleaved off. For the quassinoids, the whole side chain is cleaved off, but still both classes of metabolites have the enzymes which first modify the side chain before it's cleaved off. So maybe this means something later for the biochemistry that's happening. So there might be some relevance of that. Maybe it's just evolution, we don't know yet, but it's definitely very interesting to see that really the first 3 steps are perfectly conserved. All right, so of course we wanted to continue with the pathway and really find out what steps would be happening later in the pathway when we didn't have any additional knowledge. And as I said before, the Pearson correlation coefficients definitely had some weaknesses. So we used a different method, which is called self-organizing maps, which is something which was earlier introduced in a group of Sarah O'Connor for these co-expression analyses, which gives you a very nice visual pattern of gene co-expression. So you can see here all of these circular spheres are so-called nodes, which basically represent transcripts with a shared similar expression pattern. And these lines here in the background kind of indicate the expression pattern on average within these nodes. So here, the node which is highlighted here basically only has one peak, which in this case is root tissue. So this is basically a class of transcripts which all have very strong expression in roots. And these letters A, B, C here indicate the position of our reference genes within this whole map of transcripts, which is a very nice feature of these self-organizing maps because it's very easy to add additional annotations on top. So they are actually very convenient if you really want to see how different genes or a gene family, for example, is distributed amongst the whole transcriptome. So we looked at these root expressed transcripts in more detail, and we did some additional filtering. So this whole node contains 695 contexts, which is of course too much to screen completely. So we looked for specific keywords, looked for high enough expression in root tissues, looked for full length genes, and then in the end we removed some where we know that they couldn't be related or wouldn't be related. And we got a selection of 20 candidates, of which we could clone 18 successfully. So we went ahead with the same approach as before. We did a co-expression in Nicotiana bentamiana. Here's all the millianole biosynthetic genes, which basically gives the same peak as millianole, or pure millianole. And then we co-expressed all the P450s or all the other genes. And only one of them really showed a change in the metabolic profile. And for this CYP88, A154, we observed 3 new peaks, which of course was very nice, but on the other hand also complicated because it wasn't a clear, nice new peak. But really a mixture of 3 isomeric compounds, so all of them have the same EIC mass. So which told us that there might be some oxidation and then maybe some spontaneous isomerizations happening. So in the past, it also has been proposed that the next step in the pathway should be an epoxidation of millianole, and then millianole maybe could, or epoxy millianole could be arranged spontaneously. And to confirm or test this hypothesis, we used a chemical trick by treating pure millianole with a very known epoxidation reagent from chemistry. This is a compound called MCPBA, metachloroperbenzoic acid. Maybe some of you have heard this. This is very often used in organic chemistry if you want to epoxidize a double bond. And we were very happy to see that this basically gave exactly the same product profile. So which really supported our idea that one of these peaks could be the epoxide and the other some spontaneous rearrangement products. But nonetheless, we had to confirm. So we went ahead and purified at least here compounds 8 and 9. We haven't been able to identify this compound so far because the levels are too low. But based on NMR structure elucidation, we could really show that compound 8 here is the epoxide, and compound 9 here is a rearranged product where the methyl group moves from this position to the neighboring position, and thereby opening the epoxide ring and giving a new double bond here. This is a compound that we call isomeliandiol. This is a new natural product, but there are around 120 related tritopenoids which have the same carbon skeleton, and we call these isoprotolimonoids. This is also a rearrangement which really seems to be happening spontaneously in acid. So the purification, especially of epoxy millianole, was very tricky, and my PhD student, Chen Yu Liu, did really a fantastic job of handling this very reactive chemical. So he had to avoid all traces of acid present, and even NMR measurements in chloroform were insufficient because in chloroform, you might know there are often traces of DCl, some acid, which then also causes spontaneous rearrangement. So only when we did measurements in benzene, deuterated benzene, we really got enough stability for NMR measurements. All right, so what was still surprising for us was that we had this very complicated mixture of products. So still some unreacted epoxide, some spontaneous rearrangement product, some other unrelated, potentially shunt product. So this was a bit confusing, and we thought that maybe in the plants there could be additional factors which really channel these rearrangements into the right direction. And this is also something which has been proposed early in the literature, for example, also by Bassettli and Osborne, but also by other groups. So we proposed also that there might be some additional enzymes involved. So we went back to our transcriptome data and looked for some enzymes which might be able to do an isomerization. So we looked for keywords like isomerase, mutase, epimerase. And in the end, we only got 3 candidates, which again, we used for the co-expression experiments. And here you can see the empty vector and control reactions with isomerase 1 and 2 without the P450, which makes the epoxide. This is just the P450, which makes the epoxide and the spontaneous rearrangement products. And yeah, you can see here the product distribution for multiple replicates, which gives this very complicated profile. And we were very pleased to see that when we co-expressed the first isomerase, we got a strong shift in the metabolic profile. So suddenly we got much less of the epoxide and much more of the rearranged product, which seems to be real intermediate en route to quassinoids. So this was very nice to see, but what was even more surprising was when we co-expressed isomerase 2, we got another very different compound and a shift in the metabolic profile. So we almost exclusively got a new compound called 10 here, which was not observed by the spontaneous rearrangement before. So again, we isolated this compound and were also very surprised to see that it had a very strange and unique cyclopropane here as on top of the triterpenoid skeleton. So also this compound had not been reported before, but again, around 110 related triterpenoids are indeed known in the literature, which have some additional modifications. And this compound, which we think is this precursor of this class of metabolites that we call glubratanes, we call this one protoglubratal because we think it's the precursor of all of them. And we also expressed isomerase 1 and 2 together and could see that there was at least somewhat of a mixture between isomelliandiol and protoglubratal. Considering that both of them are expressed in the same tissues, this might indicate that really they occur at the same time and both pathways can coexist. So what are these unusual isomerases? They are actually both related to sterol isomerase from primary metabolism. This is a step which happens basically in every organism which makes sterols, so not only plants, but also humans. So you also have an enzyme called C87 sterol isomerase, or 87Si, which catalyzes a key step in sterol biosynthesis. So for example, in plants, this is an enzyme called HYD1, Hydra1, which changes the double bond from the 8-9 position to the 7-8 position. So we looked at a phylogenetic tree of these 8-7 sterol isomerases from quassinoid-producing plants, Ailanthus altissima, but also from limonoid-producing plants, which was publicly available. And we could really confirm that isomerase 1 homologs and isomerase 2 homologs occurred in all plants, which produced some of these unusual triterpenoids. So these are indicated by the full black dots here. But all other plants which only make normal phytosterols, they did not have homologs of isomerase 1 and 2. So we really think that isomerase 1 and 2 are specific to making these unique modified triterpenoids in plants and only in Sapindalis plants. So mechanistically, what's probably happening is that, of course, first we have the epoxidation, and then this epoxide can open to give the tertiary carbocation. And then what isomerase 1 and 2 do is really channel these carbocation rearrangements. So in both cases, the first step would be the same. So you have the tertiary carbocation here, methyl group shift, then you have the tertiary carbocation here, and then there are two different routes. So either the spontaneous or isomerase 1 catalyzed pathway can just be the simple elimination to give you the double bond here, or alternatively, you can basically lose a proton, and then the CH bond can also trap the carbocation to form the cyclopropane ring here, which is really in the right position to directly trap this. So this is probably what's happening. And of course, if you are familiar with triterpenoid or terpenoid biosynthesis in general, this will look very familiar because that's basically the same biosynthetic logic that is also used for the initial cyclization. So what we think is really exciting about this discovery is that this is very different from normal triterpenoid biosynthesis, because normally you first have the oxidosqualene cyclases, which make a carbon skeleton. And then afterwards, you have some additional enzymes, P450s, glycosyl transferases, which do some additional functionalizations, but they don't change the carbon skeleton. So the carbon skeleton is really made by the first step by the oxidosqualene cyclase. And then afterwards, it is not modified, but only functionalized. And what we have here is something that we call post-cyclization skeletal rearrangement, because you already have the established carbon skeleton, and then you have triggered by this epoxidase and the isomerases a rearrangement of the carbon skeleton, the methyl group shifts, and you get then the double bond or the cyclopropane ring. And this is happening after the carbon skeleton is already formed by the oxidose polycyclase. So we think that this is a very nice way to really modify triterpenoid skeletons in nature, which also kind of indicates and reiterates the normal biosynthetic logic of triterpenoids. And if you are more interested in these very fascinating natural products, for example, also the limonoids, there's also a very nice paper recently published. I guess many of you have also seen this, by the groups of Anne Osborne and Elizabeth Setterly in Science, where they look at limonoid biosynthesis and also describe the furan ring formation, for example, in limonoids, and also some biability oxidations. So yeah, I think overall there's a lot of very nice things to be discovered in these pathways. All right, with this, I come to the end. So I showed you how we discovered so far the first 5 steps of quassinoid biosynthesis. We could confirm that the quassinoid and limonoids are really related classes of natural products, despite their finally quite different looks. And I also showed you how we discovered these 2 isomerases, which catalyze the skeletal rearrangements after the initial cyclization, which basically expands the carbon skeleton diversity that we can find in triterpenoids so far. With that, I'm at the end, and I would like to highlight 2 fantastic PhD students, which who really drove this project forward. Ling Zhuang, a really great biochemist who did all the expression experiments and the biochemical characterization, and Shenyu Liu, who did a great job as a natural product chemist, isolating all these very complicated metabolites, these reactive compounds, and really managed to get very nice NMR data of all of these. And of course, thanks very much to the funding agencies, especially the DFG, German Research Foundation, and you. Thanks very much for your attention.
Thank you, Jakob. If you have any questions, feel free to put them in the chat or raise your hand and ask directly. I guess I can start with the first question. So these enzymes, I might have missed this part. The enzymes in the first 3 steps, which are similar between limonoid and quassinoid, are they orthologs of each other, or are they independently evolved cytochrome P450s that have that are involved?
Yes, very good question. Let me just go back a couple of slides. There we go. All right. So between the limonoids and the quassinoids, these are really conserved. So I forgot the exact numbers, but yeah, you can see the P450s between limonoids and quassinoids are in the same subfamily. So the first step is catalyzed by CYP71CDs. And the second step is then catalyzed by CYP71BQs. I think in both cases, the sequence identity, depending on the species that you look at, is something like 70–80%. Yeah, I think we can be pretty sure that this part of the pathway is really strongly conserved indeed, which is, yeah, in the end, not very surprising. But at least if you look from the structure, it definitely would not have been completely sure that it's like that.
Yeah, I have many more questions, but I'll let other people answer. Thanks for the nice talk.
Thank you.
Go ahead, Jack.
Hey, it's really nice talk. So I was just wondering, like, what's the expression level for the second isomerase that make Galbraithal? And then also, like, is there any other example of Galbraithal that's like being more highly modified into downstream quassinoids?
Yes, thanks very much for the question. So the expression level of isomerase 1 and 2, they are, as far as I can remember, relatively similar. So both are quite well expressed, but they don't stand out, so to say. So yeah, both are decently high expressed. But in this case, I think the crucial part for discovering these was really the chemical intuition, what could be happening, and that some additional rearrangements might be necessary, and to have the right idea about the keywords to look for. But I think if we would just have looked at the overall total expression levels, we probably might not have directly picked these. To your second question, there are definitely many more rearrangement steps happening in quassinoid biosynthesis. So we can't really be sure what exactly is happening right now. So right now it's mostly hypotheses on paper. So if you compare the intermediates that we have and the final structures, there are many more things which are happening, but unfortunately we don't have any more insights into this yet, at least nothing which is ready to put forward.
Yeah, I was wondering, like, so how many cyclopropane ring you'll see in the final quassinoids structures?
Sorry,
like how many quassinoids will have the cyclopropane ring?
So the quassinoids actually don't have the cyclopropane ring. This is really these cyclopropane ring containing metabolites. They seem to be a different unrelated class of triterpenoids, which are not truly quassinoids, because the quassinoids, they don't have the cyclopropane ring. So actually, when we look at our extracts and isolate metabolites from Ailanthus, we mostly find the normal quassinoids. We don't really see these cladribine type compounds so much, but they have been isolated before in the past, so we know that they exist. They are just a bit tricky to find. So I can't tell you how high the concentrations are, but they definitely seem to exist in the plant as well.
There is a question in the chat.
All right, let me just open the chat. Yes, so the question is if the C10 unit, which is lost or cleaved off at some point in the pathway, might have a function too, and if this can be detected in leaves. That's a very nice question. And I would love to be able to answer that, but I can't do so yet. And we are actually not even sure if this C10 unit is lost in one part, or if there might be stepwise transformations. So considering that quassinoids have some similarity to the limonoids, it's definitely also possible that we first have a transition to something limonoid-like, where first 4 carmels are lost, and then afterwards 6 additional ones are lost. Then this might also be different. But we don't really know yet the cause of transformations happening for the quassinoids. But yeah, it's a very intriguing hypothesis that also these cleavage product might have some function. But I think so far it's completely unknown how these cleaved products really look like. So what function or functionalities they would have. So yeah, unfortunately I can't answer this yet, but maybe in 1 or 2 years I would be very happy to provide you a better answer. Thanks for the question. So I don't know who was next. There's another question in the chat, how we found the isomerase candidate genes. So we actually did not find them based on orthologs from other species. Maybe I just go back to these slides. What we basically did was starting from our set of 695 gene candidates, which all had root expression, like the epoxidase enzyme, or the epoxidase gene, to be precise. And then we really looked at the annotations, for example, from PFAM annotations, and really thought carefully what enzyme could be required in the pathway, which is probably not just an oxidative enzyme, but does something different, which helps to really channel this rearrangement reaction. So in this case, based on homology, we probably would not have been successful, because this is, to the best of my knowledge, the first example that these dual isomerases occur in specialized metabolism. But here we really had to rely on annotations and on chemical intuition. And I think this would be my general advice, always to be critical about thinking what enzymes could do a specific transformation. And I think in many cases this can lead to very interesting insights. I think there are very good examples of this approach.
Benji has his hand raised.
Yeah. Hi, Jakob. Great talk. Really interesting. Good work. Talking about these enzymes, are they, do they have cofactors? Are they membrane bound? Can you tell us a little bit about how they might be working?
Yes, thanks very much, Benji. So they are really peculiar enzymes. They are extremely small. I think they are only something like 200 amino acids. They are completely embedded in the membrane. They are basically like a big filter, so to say, or a big cylinder where basically just the molecule gets in. And then there seems to be some acid-base chemistry. So there's some nice structural work on the human stereoisomerase from primary metabolism, because obviously in human sterol biosynthesis, this is also a very important drug target. What the authors have shown in this paper is that you basically just need a couple of acidic residues to transfer the proton from one end to the other end of the double bond to achieve this isomerization. And based on sequence alignments of our isomerases and other primary metabolism isomerases, I think it's probably a very similar acid-base mechanism in the end. So I think all what these enzymes do is take a relatively non-polar substrate and then put it in the right place and then use some acid-base magic, so to say, to move the groups in the right way.
Great, thank you very much.
But yeah, unfortunately, completely embedded in the membrane and very nasty to study, and also considering that there are spontaneous rearrangements. So unfortunately, the biochemistry is not very pleasant.
So I had a general question. If there, so both limonides and quassinoids are bitter, you mentioned. Do you know what sort of substructures are responsible for the bitterness and why quassinoids are 50 times more bitter?
Very good question. I'm not sure if there's really any data related to that. I think there's at least a little bit, because obviously for the limonoids, there's a big industrial relevance for citrus products. And I think it's definitely known that some chemical modifications change the bitter properties. But I think this is not really, I think this is a bit more complicated, because, for example, for the limonoids, many of them have some lactone rings in the A ring. And then I think when you open the lactone and then really get the open chain molecules, a carboxylate basically, I think it loses its bitter properties. And I think there are also some changes depending on whether you have a glycosylation or not. But I think this might not really be related to the bitter impression when you go to the taste receptors, but I think this is more some overall uptake issues. I think none of these experiments have been done in an isolated way with receptors. So I can't really properly answer this question yet, but it's a very good point, and I think also very important to understand which parts of the molecule are really related to the bitter properties.
Okay. Are there any more questions?
Oh, hey, can I ask one more question?
Mm-hmm.
Yeah. So here is showing that the OSC doesn't co-express, at least show different co-expression expression pattern with the list 2 T/50. And I was wondering, like, why do you think this is the case? Like, maybe there's like metabolite transport in the plant.
Yeah, that's a very good point. And this was definitely also quite puzzling for us, because when we started our work, we really expected that the bark, which also contains a lot of these metabolites, would be the main biosynthetic tissue biosynthetic factory. But so far, it really seems like also the roots play a big role. But then, of course, it has to imply that there must be transport mechanisms. So if you think about quassinoids in Ailanthus altissima, I think it kind of makes sense that they are in the roots, because they are typically secreted into the soil. And if you already have them in the roots, I think it's relatively easy to have a root transporter, exporter, basically, which can just get them into the soil. But on the other hand, I think many of these quassinoids also have some antiphenol activity. So then again, you must also transport them across the phloem into the bark, basically. So maybe there are some other transporters. So I think at the moment it's a bit too early to answer this question, because we don't know many other steps in the pathway. And so we can't really compare all the different expression patterns on a total level. So far, it really seems like only the OSC2 step, for some reason, is also quite present in the bark. But all of the other steps, really seem to be very specific for roots. for some reason. Maybe this also means that in the bark of Ailanthus altissima, there are maybe even yet another kind of class of tritapenoids, which are then not true quassinoids, but maybe have simpler pterocalain structures. That's definitely possible, that you already have a divergence based on the different expression pattern. But we don't really know yet. That's a good point. Thanks for the question.
Yeah, this is interesting because I think we also see the similar pattern for the lemanoids, the rutatia. Yeah, I'm one of the authors on that science paper. So yeah, I think the OSC also doesn't co-express well with all the downstream enzyme. And yeah, we also don't know why is that the case.
Yeah, I think it might be really a possibility that there are other unrelated non-lemanoid, non-quassinoid tritapenoids, and then this really just uses the same precursor. Why not? I think it could make sense. And I think it would fit the data quite well.
Okay, if there are no more questions, please join me in thanking Jakob for this wonderful talk. And
thanks very much, everyone, for joining in.
Hope to see you all in the next session. Thank you.
Yes, hope to see you too. Thanks very much.